So Dr. Navarro studied in Paris, but he also did his PhD at the Pasteur. After graduating, he remained at the Institute as a research associated, working with Professor Avner on chromosome X activation. And then he moved to the University of Edinburgh to work with Professor Ian Chambers on pluripotency regulation in stem cells. Shortly after, he returned to the Pasteur as a junior group leader after winning several grants and many awards. And then he first became coordinator of the Revive Bioinformatics team, and then the head of the unit of epigenomics, proliferation, and identity of cells, or in brief, the EPIC unit. So we are very happy to have you here today, and the floor is yours.
Thanks, Matteo. Can you see my screen?
Yes, that's perfect.
Okay, perfect. So thanks a lot for the invitation, but more generally, actually, to have it set it up such a nice initiative for the community during this period. So the work I'm going to discuss today with you is about our trials to understand how transcription factors behave during mitosis, and in a few words, this can be really summarized with the title of this talk. What transcription factors need to decide during mitosis is whether they stay or whether they go outside of the chromatin, and perhaps more specifically, the question that we like to address is whether when they stay on chromatin, they just stay like the two guys here on the back of the stage, just dancing around the chromatin, or whether they really engage some of their energy to do something more significant for gene regulation. So before I start, I'd like to acknowledge two of the major drivers of this project over the years. The first one, Nicola, who started very early with me as a postdoc, and I was very lucky that he stayed in the lab as a CNRS permanent researcher now. He really spearheaded all of our work on mitosis. And on the other side, people who went away after the war, we have Nick Owens, who was really instrumental to set up all the computational pipelines that we are still using today. Of course, these two guys didn't work alone. They've been helped by a number of people, and just to mention some of them that did an important contribution for the work I'm gonna describe today. We have Inma, who is now a permanent researcher here in Pasteur, a postdoc, Amandine, two computational scientists, David and Almira. And for people that went away, we have Taleya, who was really an extremely talented experimentalist who is now in the private sector. So I'd like to start this presentation just by trying to give a very broad introduction. So what we are trying to understand is how gene regulatory processes cope with mitosis, with the purpose of efficiently reproducing gene expression patterns in daughter cells. So as you all know, the real purpose of mitosis is to equally distribute the genetic information between the two daughter cells. But this really comes at a pretty high cost. A cost for gene regulation, which is labeled here in red, but more widely, a pretty important cost for the cell itself, and in fact, even for the tissue. So just to give you a few words, mitosis is driven by a very intense phosphorylation cascade that will not only inactivate mitosis, but start mitosis, sorry, but inactivate specific transcription factors, either inactivate them or in some cases even induce their degradation. This is accompanied by a global transcriptional silencing, the condensation of the chromosomes, as you can see in this picture here, and the breakdown of the nuclear envelope. So what we try to think about is how the cells transit through this stage in order to nevertheless convey some regulatory information to the daughter cells. So we can think of two different sets of mechanisms. On the one hand, we can think of passive mechanisms. Transcription factors and other gene regulators are simply cytoplasmically inherited, and given the robustness of gene regulatory networks, this might be largely sufficient to reinstate some of these requirements. We can also think of all these systems of chromatin-based inheritance, all these modifications that are instructed by transcription factors during interphase, but when they persist throughout mitosis, they can nevertheless convey gene regulatory information to the daughter cells. What we are mostly interested in contrast are what I would like to call active mechanisms, transcription factors that remain active during mitosis, not only floating around the chromatin, but actually engaging on site-specific interactions with at least a subset of the targets they control during interphase. And these are the transcription factors that I like to refer to as mitotic bookmarking transcription factors. So we are trying to address these questions in the context of mouse embryonic stem cells, which, as probably most of you know, are undifferentiated cells derived from early embryos that have a number of particularities that I think make them a very suitable model system to study how mitosis influences gene regulation. To start with, these cells self-renew, which means that you can take single undifferentiated ES cells and they will grow as small colonies first, then further expand, and you can repeat these procedures virtually infinite numbers of times. And this means that these cells can really transit through mitosis almost endless without dramatically affecting their identity and their developmental potential. Another particularity of these cells is that in contrast to somatic cells that typically have a very long G1 phase during which they can, in a very relaxed way, reinstate gene regulation appropriately, ES cells do not have a G1S checkpoint. And this is a big issue in reality because this free pass from mitosis to replication essentially makes those cells alternate between mitosis, which is an issue for gene regulation, and replication, which is another issue for gene regulation. And nevertheless, they are extremely good at preserving their molecular identity throughout these phases of mitosis and replication. The last aspect that I think is very important to study this subject is that all these phenotypes here are strictly driven by transcription factors in ES cells. You probably know NANOG, OCT4, and SOX2 as the main pluripotency transcription factors, but there are many others, in particular these orphan nuclear receptor, ESRB, which stands for estrogen-related receptor beta, and we are gonna be speaking a lot of these transcription factors. So if we now try to see a bit how these factors behave in mitotic cells, the first impression is that there is nothing really interesting. Here, each transcription factor is labeled in green, and as you can see in these mitotic cells, they are fully excluded from the mitotic chromosomes, which are labeled here in red with this Ki67 protein, which is at the surface of each of individual mitotic chromosome. But as you can see here, this is upon fixation with formaldehyde. And for those of you who have been following this seminar series, Sheila Tibbs already described last year that when transcription factors are fixed with formaldehyde, they are artificially evicted from the chromatin. Indeed, what we see by live imaging is that even though some transcription factors like NANOG are also evicted from the chromatin, the others may have very different behaviors, ranging from very interesting patterns that we can discuss after for OCT4, which is nevertheless excluded from the chromosomal arms seen here in red, to factors like SOX2 that really have a very focal enrichment on the mitotic chromosomes. And we see something very similar for ESRB, which you can see decorates here each individual chromosomal arm. So this is obviously very important because on the one hand, it has been a very strongly confounding piece of information over the last 30 years, pushing the idea that during mitosis, everything is dead. This is probably artificial. And on the other hand, it is a technical issue to work with these factors in mitotic cells. So we expended a lot of time during the very first years of the creation of this lab to try to identify alternative cross-linkers. In this case, you can see D-succinyl glutaraldehyde, which would allow us to fix in appropriate ways the behavior of transcription factors in mitotic cells. And as you can see here, NANOG is excluded, OCT4 displays this unusual pattern, and SOX2 and ESRB are fully fixed within the mitotic chromosomes when we combine D-succinyl glutaraldehyde with formaldehyde. So this is important because it gives us a very strong technical way and very simple to really address where these factors are binding in mitotic cells. So we can do now ChIP-seq in very good conditions. And as you can see, among these 4 pluripotency transcription factors, only ESRB retains substantial binding throughout the mitotic chromatin. Even SOX2, which is very good at coating these mitotic chromosomes, is almost completely evicted from its interphasic targets. And this is most likely due to the fact that SOX2 is specifically phosphorylated in mitosis to inhibit its capacity to recognize its DNA binding motif, but this is a work from other labs. So now that we have established that ESRB does behave as a canonical bookmarking factor, engaging in site-specific interactions with at least a subset of its targets, the big question that I would like to address with you today is what does it do? Our first idea is that it would be just passively staying on the chromosomes. At the end of the day, maybe the cell doesn't need anything else than that, someone guiding how the genome has to be reactivated in interphase. However, both by FRAP and by FLIP, you can really appreciate that very rapidly after photobleaching half of this chromosome here, the signal comes back. It's almost immediate. Conversely, you can photobleach at the 2 poles of the cells here, and in less than 10 seconds, the coating of the chromosomes completely disappears. So clearly, these factors are not just bound to the mitotic chromosomes, they are extremely dynamic. The second idea we had was also very naive. During mitosis, we believed the chromosomes were fully condensing. So simply we thought that the binding of ESRB would preserve local accessibility at a subset of binding sites, perhaps promoting gene reactivation in the daughter cells. However, when we address the level of accessibility at a number of regulatory elements, promoters, enhancers, and those regions that are bound by ESRB in interphase and mitosis that we like to call the bookmarked positions, or regions where ESRB is evicted in mitosis, and addressed by ATAC-seq the level of accessibility, as you can see, there is not a big difference between interphase and mitosis. It is true that there is a small decrease at lost positions, but even at bookmark positions, we see some minor reduction. In fact, the big surprise in this field, and George Blobel's lab was really the first to show this, is that genome-wide chromatin accessibility is quite largely retained in mitotic cells. This was very shocking for us for 2 reasons. First, because this is not the way we expected mitotic chromosomes to be. And second, because if everything remains accessible like this, who cares of mitotic bookmarking of ESRB? At the same time, does that mean that every peak here, essentially all peaks that are accessible in interphase and that remain accessible in mitosis are bookmarked by a transcription factor? I don't think that this is really possible, although it's very difficult to prove against that idea. But what I really want to point here is that, okay, enhancers, for example, in this case, do maintain significant accessibility in mitotic cells. But what does that mean? Does that mean that nothing is happening at these chromatin sites? Well, if you now, instead of measuring accessibility, you measure where the nucleosomes are specifically located in mitotic cells using MNAs, what we can observe is that this beautiful nucleosome depleted region here in interphase in blue, where transcription factors are binding in interphase, gets completely refilled by nucleosomes in mitotic cells. So our interpretation here is that most transcription factors are inactivated by the mitotic cell, and their eviction from the chromatin allows the nucleosomes to invade these sites. And this is important for the daughter cells, as they will require extensive chromatin remodeling to reopen these regions and allow proper transcription factor binding. At the same time, it was interesting because ESRB, because in that case, we have a window of opportunity, let's say, to allow ESRB to change something to how the chromatin is configured in mitotic cells. So to address this, we need to come back to our sites, which are bound by ESRB in interphase here, and only at a third or so of those sites in mitosis. The first thing I want to say is that those sites where mitotic ESRB binding occur tend to be regions that have extremely good ESRB motifs. In fact, these heat maps here are ordered by mitotic ESRB binding. And as you can see that we have an almost perfect correlation with the quality of the ESRB motif that we can identify in those regions. Below a certain threshold here, ESRB is no longer capable of interacting with this chromatin in the context of mitosis. So if we now look at the nucleosomes over these bookmark positions, what we see is that the profiles in interphase and in mitosis are very similar. In fact, this small nucleosome depleted region here at the center, all this is centered on the ESRB motif, so presumably ESRB is going to be binding here is perfectly preserved in mitotic cells, and these beautiful nucleosomal arrays are also fully maintained in mitosis. We then needed to look at, let's say, our control, which is whether at these regions here where ESRB is going away in mitosis, the nucleosomes completely lose position and invade the small nucleosome depleted region made by ESRB in interphase. However, what we observed was very, very surprising. Essentially, at those regions, we have no order for the nucleosomes. They seem to be randomly located throughout these regions. But then remember that these regions, they are all centered on the ESRB motif. And I just told you that the ESRB motif here is really poor. So we started looking at other motifs that could be more enriched over these regions. And what was very obvious is that the OXOX motif has nearly an opposite pattern to the ESRB motif. If the ESRB motif is high at the bookmark positions and then slowly decreases down, the OXOX motif that we find over these regions shows exactly the opposite. It is very low at bookmark positions and then progressively increases. And this completely matches the patterns of OCT4 and SOX2 binding that we see by ChIP-seq in interphase for these two transcription factors with high levels at those regions where ESRB binding in mitosis is very low, if existent at all. So our interpretation of this data is that at the bookmark positions, ESRB is really the driver. It binds here because it recognizes its motif, and it recruits its friends, OCT4 and SOX2. At these positions, ESRB is able to recognize this very good motif also in mitosis. In contrast, at all these regions here where ESRB is lost in mitotic cells, in fact, ESRB is not the driver. The driver is OCT4 and SOX2. Those are in fact OCT4 SOX2 binding sites, not ESRB binding sites. And ESRB is a passenger here. Since these two factors are evicted during mitosis, then ESRB is not able to come to recognize this poorly conserved ESRB binding motif. So what was really interesting is that when we took this set of regions here, which are centered on ESRB, and we simply recenter them on the OXOX motif that we identified, we recovered a very decent nucleosomal order with this very strong nucleosome here in interphase right on the OCT4 SOX2 motif, which you may know act as pioneer transcription factors capable of engaging in base-specific contacts with their motifs, even in the context of a nucleosome. In mitosis, when these factors go away and ESRB goes away, the nucleosomes largely reconfigure their positions throughout this central region. So our idea really is that the main function of ESRB during mitosis is to keep nucleosomal order at its binding sites, at least at those where there is a very good ESRB motif. The question I want to discuss now is whether ESRB is doing this alone, or whether it is doing this with friends, and if so, with which friends, because its canonical co-binders, NANOG, OCT4, and SOX2, are evicted from the chromatin in mitotic cells. So to address this, we use the targeted proteomic approach, where we fix the cells in either interphase asynchronous cells or in mitosis with DSG and formaldehyde. And we perform an ESRB IP followed by mass spectrometry. What you can see in this plot here on the right are proteins that are enriched in the ESRB IP versus a control IP. On the left, you have the same thing, but for cells that have been purified in mitosis. And then on the y-axis, you have the statistical significance. So essentially in this blue square, we have the factors that are identified with ESRB in asynchronous cells. And in this red box here, you have the proteins that are identified with ESRB in mitosis. So what we can see already is that the density of dots here is much higher than in mitosis. Perhaps 3 to 4-fold more proteins are identified in asynchronous cells than in mitosis. So there is a reduction of the number of proteins that bind the chromatin with ESRB. If we put some names on these dots, obviously you have the pluripotency factors SOX2, OCT4, and NANOG, which are detected in interphase, and in mitosis they go to the backbone. Then we have two major nucleosome remodeling complexes that were already known to interact with ESRB, SWI/SNF, and the NERD complex. And here in yellow, you have some of their key components. Most of them also go away with the exception of BRG1 and CHD4, which are two key enzymatic activities of these nucleosome remodeling complexes. And this, I think, might be important for the nucleosome-related activity of ESRB in interface, but also in mitosis. The final points that I want to highlight from this proteomic analysis is that we have a large family of proteins shown here in red, which are detected both in asynchronous cells and in mitosis. And this is the only single family that behaves as a family associated with ESRB, both in interface and in mitosis. And it turns to be that those are either nuclear receptors like ESRB, and this is the case of its very close relative estrogen-related receptor alpha, and two other nuclear receptors which are very close to ESRB, NR5A2 and NR0B1, and the retinoic acid receptor, which is very well known from the pluripotency field. And then we have four of the known cofactors of nuclear receptors, not necessarily in ES cells, but also in other contexts. So I find this quite actually surprising that nuclear receptors really seem to associate with ESRB. It perhaps makes sense because all these factors here, they are structurally related, and they also share the same DNA binding motif with some distinctions. So we were very pleased to see NR5A2 in this list here because Nicola was already working on this factor. In fact, what he showed is that while it is true that the deletion of ESRB is not fully penetrant in ES cells, there is a phenotype, but it is not dramatic. When the two factors are removed, ES cells cannot any longer self-renew. Moreover, in asynchronous cells, these factors tend to bind together. And as I said, they share the binding motif with a small exception. ESRB prefers a T at this position, while NR5A2 prefers a C at this position. But the differences are, in fact, quite minor, at least when we compare their binding intensity to regions that do not have a motif for ESRB or NR5A2. So we were obviously very interested in trying to follow the activity of NR5A2 in mitotic cells. And for this, we performed chromatin IP after DSG and formaldehyde fixation. We can very easily make these four clusters of regions where both ESRB and NR5A2 are binding both in interphase and in mitosis, regions where the two factors are binding together in interphase, but evicted together from mitotic cells, and then two sets of regions where either ESRB is the only bounder and bookmarker, or regions where both ESRB and NR5A2 are binding in interface, but only NR5A2 and mitosis. From these regions, the ones that I find particularly informative are those here, these regions that are bookmarked exclusively by NR5A2, because before knowing the existence of these regions bound by NR5A2 in mitosis, they were assigned to regions that lose ESRB in mitosis. So the real question here is whether at these regions, the order of the nucleosomes is maintained in mitosis or whether it is completely gone. So when we address these three major clusters here, bookmarked by the two factors, bookmarked only by ESRB, or bookmarked only by NR5A2, you can see that the profiles are relatively similar. The first conclusion, which is important, is that over these three clusters, the nucleosomes are phased. They are phased according to the ESRB NR5 motif, which is right on the middle. And we don't see any major invasion of these regions by nucleosomes in mitotic cells, which makes sense because the three clusters are bookmarked by at least one of these two factors. The other noticeable observation is that perhaps NR5A2 has a stronger impact than ESRB itself when we look at these profiles. The NDR, but this is also true in interface, is, let's say, better over these regions than over these regions here bound exclusively by ESRB. But clearly the two factors work together, as the nucleosomal profiles that we observe here are really, really clear when the two factors are binding together in interphase and in mitosis. So the conclusion from this is that indeed the two transcription factors can organize the nucleosomes in interphase and in mitosis, but when they are together, they do it better. So to try to finish over the last few minutes, I'd like to share with you some of the thinking we are following to try to find the function a gene regulatory function to these events. And this is really the less trivial part of our work. So the experiments I want to share are based on this very simple setup where we arrest our cells on mitosis and then we release them to re-enter into the cell cycle up to 2 hours. And you can see that the chromatin is progressively decondensing. So we performed RNA-seq to address pre-mRNA levels using a specific pipeline. And in this PCA, what you can see is that from asynchronous cells here, you go here to mitotic cells, and then you have something that I was initially very surprised, which is that instead of rapidly coming back to the average, the transcriptome further diverges from the asynchronous, up to a point that the most different transcriptome that we have in this kinetics to the average of asynchronous cells is obtained after around an hour post-mitosis. These cells here have a transcriptome which is more different to asynchronous cells than mitotic cells themselves. And then the transcriptome progressively comes back. So the reason for this, I believe, is because, in fact, when you look at the loadings of this PCA and you try to find the most important genes contributing to these variations, what you see is essentially the cell cycle, either generic cell cycle or G1 and G1S transition phases, or even the S phase. And remember that these cells are special because they have a very short G1. So very rapidly, they need to rebuild all the machinery to enter into replication. So most of the variability in transcriptional activity that we see here is in fact driven by a major requirement of the cells, which is to enter into replication. And this is obviously a very confounding effect to try to find where ESRB and NR5A2 are controlling gene reactivation dynamics. The second component of these dynamics here is not the cell cycle. It is the fact, and this is something that Gerd Blobel already showed, that there is a very intense and pretty global burst of transcriptional activity just after mitosis. So regardless of the level that we see in asynchronous cells and how much it goes down in mitosis, globally, the genome really has a burst of transcription that goes well above the levels that we see in asynchronous cells and then starts to go down. Again, in the context of this global burst, I believe it's going to be quite difficult to identify the role of these mitotic bookmarking factors unless they are controlling these aspects, which is not impossible. And the final point is that despite these global trends, gene reactivation is quite heterogeneous across genes. You have genes like KLF4, which really has a very dramatic burst within an hour and then goes down quite dramatically. You have genes that have a slower dynamics and then remain more stable, and genes that spike at around 1 hour and then go down, like very important genes such as OCT4 and NANOG. What was also quite interesting is that genes that are not really detectable in asynchronous cells, they undergo this spike, and these can be genes which are in fact not really highly expressed in ES cells, such as EOMS. So to try to put some order here, we simply ranked our gene reactivation dynamics by the reactivation strength. And as you can see, as we go down through this heat map, we enter into portions of the genome which are more highly and more rapidly reactivated after mitosis. So if we now ask the question of what part of this heat map here is statistically associated with those sites that are bookmarked by ESRB and NR5A2, what we see is these results here. At the top of the heat map here, there is very small association with our ESRB and NR5 bookmark positions, and even with the lost ESRB positions and the NR5-2. What we see when we go down is that we start increasing this statistical association up to a point here where the transcriptome is really reactivating strongly. And here we have a dispersion of our statistical significance with the regions that lose ESRB in mitosis displaying relatively low levels, and only the regions that are bookmarked by ESRB and or NR5A2 gaining statistical significance in terms of the number of peaks that are found within 100 kb of the promoter of all these genes. Of course, you can argue that this is biased because those genes are more highly expressed in asynchronous cells. So what we are spotting here is just a correlation between strong transcription factor binding and strong transcriptional activity. So to address this in the last 30 seconds, I would like to say that we have put some efforts in trying to identify groups of genes that have a very specific post-mitotic transcriptional patterns. We have genes that are expressed in interphase here, they go down in mitosis, and then they reactivate either very strongly and very rapidly here in red, or almost as rapidly, but with less intensity here in purple. And we want to compare these to genes that are not really expressed in asynchronous cells or at very low levels or early during G1 in reality, but that nevertheless reactivate very rapidly after mitosis, and they reach a level which is very similar to these Onslow genes. When we now compare the statistical enrichment of our chip clusters to these three groups of genes, what you can very rapidly appreciate is that no matter the distance at which you want to do this analysis, the genes that reactivate slow after mitosis have very poor association to our regions. In contrast, those that reactivate fast, either being highly expressed in interphase or being poorly expressed in interphase, they have a very strong association with our ESRB and NR5 peaks. And this is particularly true in terms of the distinction of bookmarked here in color to lost positions here in black for these genes that in fact are not even expressed at high levels in interphase. They are characterized by a fast reactivation dynamic after mitosis. So just to conclude is difficult because it's difficult to reach conclusions in this field, but at least to tell you what I find important in all this data. The first thing is that there are several modalities of interaction between transcription factors and mitotic chromosomes. You have transcription factors that are fully evicted. You have transcription factors that coat the chromosomes, but without really engaging in site-specific interactions. And then you have what I like to call proper mitotic bookmarking factors that engage in site-specific interactions. Another important point is that chromatin accessibility and nucleosome positioning are not necessarily the same thing, at least in the context of mitosis. And what mitotic bookmarking factors seem to do is really to control nucleosomal order in mitotic cells. And then also, something which is very important is that during mitosis, there is something that I like to call the regulatory reduction. So it's not a genetic reduction, obviously, this is more for meiosis, but in mitosis, what we see is that there are less transcription factors that can engage in site-specific interactions. These transcription factors restrict their binding to regions with particularly good motifs, so they bind less regions. At these elements, they recruit less proteins, with perhaps the exception that nuclear receptors behave as very general bookmarking factors. And finally, unfortunately, we can still not conclude about the function of these factors, but what I want to highlight is that this might be very difficult because after mitosis, in fact, the cell is busy with the cell cycle and with the reactivation of the whole genome. So finding the specific responses to bookmarking factors is going to be difficult, but to address this, we are currently generating cells where we can rapidly inactivate at the same time ESRB and NR5A2. The issue is that maybe there are other nuclear receptors which are also binding at the very same regions. So to finish, I would like just to thank the whole lab. I already highlighted the key people. Among our collaborators, I would like to really highlight Petra and Irem for their help with proteomics. And thanks again for the invitation and for your attention.
Thank you very much, Pablo. Great talk, very, very, very elegant study. Just before we move into the questions, I already see a couple coming. Just a reminder that if you want to talk with the speaker, just let us know in the chat so we can keep you in the meeting afterward. We have Andrew Oldfield. Great talk, Pablo. Just curious how you think SSRB bookmark sites are even more nucleosome depleted during mitosis than in interface?
Yes, thanks, Mr. Vieuxon. So I think the answer, do you still see my screen or I blocked it?
Yes. It's visible.
It's visible, okay. So what Andrew is asking is why in mitosis the nucleosomes are in fact changing a bit at ESRB sites. the NDR is better or apparently better. And in fact, there are other movements that we can see here. In fact, they slightly roll versus towards the motif. What we find out, and this was before we understood that other nuclear receptors were also binding these regions, is that while these regions are centered on the best ESRB motif, when you look at the second best motif for ESRB across these regions, you see a very strong enrichment in the center, but also at this position here and at this position here. So what we believe is happening is that at these sites, ESRB is the driver in interface, but in interface, it is still capable of recruiting a number of proteins, such as OCT4, SOX2, NANOG, and probably many others, which also have their say in terms of nucleosome organization. So they will not necessarily bind within the NDR. Maybe they will bind on this nucleosome, or maybe they will bind here. And this will impose additional constraints to the way these nucleosomes are ordered. In mitosis, there is this mitotic reduction. All these proteins go away. Only nuclear receptors bind here, so it will be purely genetically driven. They will be binding here, they will be binding here, they will be binding here, and they will organize the nucleosomes exactly at their will.
Great. Then we have Andrea Bernardini. So beautiful talk, just a curiosity. In the slide in which you show the nucleosome profile in asynchronous versus mitotic cells, it seems that there is a slight shift of nucleosome peaks away from the origin of the plot in mitosis. Is this due to the addition of a central nucleosome that pushes nearby nucleosomes away from the central site?
So this is exactly,
exactly. That you just answers now. Yeah, indeed. Then we have one more from Sandeep. Sandeepan, great talk, thanks. Are you able to share some thoughts on how will you reconcile the accessibility versus nucleosome positioning disconnect at the bookmark sites?
This really drives me mad. I have, really, for me, it's the most important observation that we and others have made in this context.
Yeah.
Why regions that lose transcription factor binding in mitosis remain nevertheless accessible? It's something that is complicated. I mean, ideas are free, but they have no value until you have any data to put on them. So we can share ideas. One idea we had. So let me say something first. I think for this we need to distinguish promoters and enhancers. Let's say. So at promoters. You heard Sheila's talk last year. If TBP is really binding all these promoters, then the fact that they remain accessible makes sense, at least in ES cells. For enhancers, I think it's more complicated. One idea that I would like to push, but we have no data yet, is that maybe mitotic proteins that are required to make mitotic chromosomes hijack regulatory elements to load into the chromatin. So this is something that I like very much as an idea, but again, we have no data. So this would give an impression of accessibility, but distort the way the nucleosomes are positioned.
Yeah, no, that's great. It's really fascinating the whole nucleosome part. There's a last question. Seems a curiosity by Nancy Gutierrez Hernandez. She's asking about OCT4 in centromeres. If you know anything.
So I have no idea except that it's what we see. So both in live and by DSG fixation, we do see a very focal enrichment of OCT4 over chromocenters in interface, and in mitosis over centromeres. There are other transcription factors which have been shown to behave like this, PAX3 or PAX6, I don't remember which PAX actually, but it's not the first transcription factor that behaves that way. Why this would be important for ES cells? I mean, OCT4 is a hugely important transcription factor for these cells. So how would it behave that way? I can't tell. I'm not gonna risk giving a very wrong idea.
Very fair. Just last question popped up. SSRB can play a role of mitotic bookmarker because of a nucleosome arrangement in the genome. You show that you show data that some of their motifs are exposed in mitosis as it is an interface.
I'm sure I got there is a question there. Maybe
yeah,
more than a question.
So I think is more about the rearrangement.
Can I read it somewhere, or is not even? I don't see it.
Yeah. It's so if I can rephrase it, probably can there be play a role as mitotic bookmarker because the nucleosomes are rearranged in the genome. So I guess it comes a bit on the chromatin remodeling side of the aspect.
It's inverting the question somehow. Is it ESRB driving the order of the nucleosomes, or is it ESRB binding there because the nucleosomes are organized in a specific way? And at the very beginning, this is what we thought when we saw the nucleosome positioning over the regions that lose ESRB in mitosis being completely random. Our first impression was, gosh, we're wrong. In fact, it's the nucleosomes which drive binding and bookmarking. The fact that we were able to find order over these regions just by shifting the perspective from ESRB to OCT-SOX really suggests to us that it is really transcription factor binding which will have a strong say in how the nucleosomes are ordered across these regions. And then obviously we have all these nucleosome remodelers that interact with ESRB in interface, much less so in mitosis, but this is something we need to follow up.
Okay, great. Perfect. So if there are any questions, please join us in the coffee session. We'll start briefly. Let us know again in the chat if you want to join.
Thank you very much, everybody.
Yeah, talk again. Thanks you both.